Hi guys, today I'll be presenting our research on the RS1800629 SNP in the TNF gene and its role in rheumatoid arthritis. RA is a chronic autoimmune disorder characterized by persistent inflammation in the joints, leading to pain, swelling, and eventual joint damage. While environmental factors such as smoking or infection can trigger RA in genetically predisposed individuals, genetic susceptibility plays a significant role in the disease development. Genome-wide association studies, or GWAS, have identified several SNPs linked to RA, many of which reside in non-coding regions of the genome. These non-coding SNPs may influence gene expression by altering regulatory elements, making it challenging to understand their exact role in the disease process. The SNP we focus on today, RS1800629, is located in the promoter region of the TNF gene, which encodes tumor necrosis factor alpha, a key pro-inflammatory cytokine involved in RA. Elevated TNF levels contribute to inflammation in RA, and TNF inhibitors are used as a therapeutic strategy to reduce inflammation in patients. The methodology we used in our study is based on the framework presented in a 2024 paper, which integrates GWAS findings with functional genomics to understand the role of non-coding SNPs in diseases like RA.
Let's now go over our objectives and hypothesis. Our primary objective is to investigate the functional role of RS1800629 in modulating TNF expression in RA or rheumatoid arthritis relevant tissues, and more specifically in immune cells such as T cells and macrophages. These immune cells play a central role in the inflammatory processes that drive RA. Our hypothesis is that RS1800629, located in the TNF gene promoter, increases TNF expression in these immune cells, leading to a more pronounced inflammatory response in patients with RA. We believe that this SNP influences TNF levels, contributing to the chronic inflammation observed in RA. To test this hypothesis, we will use a range of computational tools to map the SNP's exact position within the promoter region of the TNF gene, explore its potential effects on TNF gene expression, and understand how this SNP may be involved in regulatory pathways that contribute to the pathology of RA. This approach is directly inspired by a 2024 study that emphasizes the integration of GWAS, or genome-wide association study findings, with functional genomics to identify how non-coding SNPs may impact gene expression and disease development in diseases such as RA.
Now we are going to talk about the methodology. Our approach follows the framework that was presented in 2024 paper, which successfully integrated the GWAS data with functional genomics to explore genetic variants that contribute to complex diseases such as RA. Following this approach, we used several computational tools to analyze the RS1800629 SNP. First, we mapped the SNP's position within the TNF gene promoter using the UCSC Genome Browser, a tool that provides detailed genomic visualizations. This allowed us to identify regulatory regions near the SNP that may influence gene expression. Then we used Ensemble to explore how the RS1800629 connects to the TNF and other genes involved in inflammation. Finally, we used GTEx database, which provides gene expression data across different tissues to analyze TNF expression specifically in immune-related tissues such as T cells, macrophages, and synovial tissue, all of which are central to RA inflammation. By using this methodology, we are able to understand whether RS1800629 influences TNF expression in these tissues and how it may affect RA progression, which aligns with the functional genomics approach outlined in the 2024 paper.
Let's look at the key findings from the results. Figure 1 shows the genomic position of RS1800629 in the TNF gene promoter, highlighting its location in a regulatory region that likely modulates TNF expression. Figure 2 illustrates nearby structural and sequence variants that may interact with RS1800629, collectively influencing TNF regulation in immune cells. Figure 3 presents EQTL violin plots, showing a strong correlation between RS1800629 genotypes and TNF expression levels, with certain genotypes linked to higher expression. These findings support the hypothesis that RS1800629 impacts TNF expression in immune tissues, contributing to inflammatory processes in rheumatoid arthritis. This analysis builds on the 2024 study, leveraging EQTL and GWAS data to connect genetic variants to gene expression. In conclusion, our study combines GWAS data with functional genomics to investigate the role of RS1800629 SNP in the TNF gene promoter and its contribution to inflammatory processes in rheumatoid arthritis. Through using tools like the USCS Genome Browser, Ensemble, and GTX, we analyzed how non-coding variations like RS1800629 regulate TNF expression in disease-relevant tissues. Our findings indicate that SNP impacts TNF expression in immune cells, offering valuable insights into RA pathogenesis. This integrative approach is critical for understanding the complex genetics of autoimmune diseases, paving the way for identifying biomarkers and therapeutic targets.